Good afternoon. It's my pleasure to introduce the speaker of today's LIVA seminar of the CIBER. And Professor Schalk van der Merwe obtained his medical degree at the University of Pretoria in 1991 and completed his residency in internal medicine and thereafter his fellowship in gastroenterology in 2000. And thereafter he moved from South Africa to Europe, and he did a PhD at the Free University of Amsterdam. And thereafter he came to Belgium, where currently he is an associate professor of medicine in the Department of Gastroenterology and Hepatology at the University of Leuven since 2011. And in Leuven, he heads the Laboratory of Hepatology in the Department of Chronic Diseases, Aging and Metabolism at this university since 2017. And in this laboratory, he maintains a very intense research activity in the pathology and more in depth in the mechanisms involved in cirrhosis-associated immune dysfunction, in the dysfunction of the innate immune system in cirrhosis, in decompensated cirrhosis, and in ACLF, and trying to understand the mechanisms involved in this dysfunction and its contribution to the susceptibility of these patients to develop infections and to the progression of cirrhosis in itself. And it's because of this research that for a long time we became close friends, and because we are involved in this in the same research activity. But it is also important to say that a hidden aspect of Schalk professional activity is that he is also responsible of as an hepatology of the advanced hepatobiliary and pancreatic interventions in the hospital at the University of Leuven. And in this settings, he performs or participates or is the head of a group that performs more than 1,700 ERCPs and more than 1,000 therapeutic endoscopic ultrasounds interventions yearly. I remember when I visited him years ago, and he showed me the endoscopic suite. And I told him, well, we do quite a lot of endoscopic ultrasounds in our unit, maybe around 450 yearly. And he told me, well, we do around 1,200 every year. So I said, wow, and it's an incredible number. Schalk, thanks for being here today. This seminar had to be canceled last year when it was scheduled for mid-December. Thereafter, COVID is among us, and we know the effort that you are doing to be with us, not in person, but we appreciate your time, since we know that you have a very high clinical load and the compromise of the routine clinical duties because of COVID. Thanks, Schalk, for being here, and our pleasure to have you here.
Thank you so much, Augustine, for that very nice introduction. And thank you also to Rafa and Jordi for the invitation. It is indeed, I'm very humbled by this because I've always regarded Spain as certainly the leading country in the world when it comes to liver research. And so it's a very humbling experience to share with you what we have been doing in the lab as well. And hopefully it will strengthen ties with you further with regards to research in the future. So as I was introduced to speak about the role of monocytes and macrophages, and I will start off by showing you this slide. Certainly when I went to university, we were brought up by the concept at that time that macrophages are replenished from circulating monocytes that originate from the bone marrow. And recently, the concept has evolved, where we now understand that macrophages populate from yolk sac or from fetal liver tissue sites during embryology, during development, and then self-renew from these tissue sites over time. And only when there's damage will there be an influx of circulating monocytes to replenish macrophages that are lost through disease. This has certainly been the case now for the last while, and from evidence that was generated in our lab as well, there's now evidence that intestinal macrophages may also be part of this group. Previously, it was thought that intestinal macrophages will only be repopulated when they're lost from the circulation, but some evidence generated now suggests that at least partly the intestinal macrophages repopulate locally, and over time, maybe as the animal ages, will macrophages, monocytes originating from the circulation, also enter the intestine. So with this knowledge, I think this has really a few years ago sparked our interest into how are these macrophages, or these populations of macrophages, involved in liver disease in the liver, but also upstream to the liver, in the intestine, in adipose tissue, as well as monocytes in the circulation. So I think everyone attending this webinar will be very familiar with this concept that cirrhosis should be seen as an immunosuppressive state, as these patients, as patients with cirrhosis are very likely to be admitted to hospital. When they are in hospital, they're very likely to develop new infections. And when this happens, they are very prone to develop organ failure, which increases in-hospital mortality and their survival if they leave the hospital. Now, Augustine published this a while ago, and I think at that time, in 2014, it really crystallized very nicely the knowledge at that time regarding how the situation evolves as patients progress from a normal situation to compensated cirrhosis, and then on to decompensated cirrhosis. And we now, because of this publication, we can now understand that as decompensated cirrhosis develop, that the normal effector responses of monocytes change. And I will come back to that in my talk, to the extent that patients may become more susceptible to bacterial infections when a certain bacterial threshold is achieved, and under which those patients are prone to infection, recurring infection, which precipitates ACLF. So the fact that ACLF should be seen as a immunodeficiency state is very important. And in my talk, I will focus on why we think this is happening and what the role is of immunodeficiency and systemic inflammation. So I want to propose to you that we should now see cirrhosis rather as a multi-systemic disease, and not just as a liver disease, based on the fact that in all tissue compartments in cirrhosis, including adipose tissue in the small intestine, certainly in the liver, but also in the peripheral circulation, the environment changes. And as the environment changes as a result of cirrhosis, immune activation occurs that will add to chemokines, cytokines, as well as changing immunosuppressive cell populations being generated and released in the compartments, as well as in tissue sites that will affect the function of the immune response, the effective function of the immune response to infections. In the liver, this is very well known to all of you. As cirrhosis develops, Kupffer cells will change from TIM4 CD68 expressing cells to cells that have been altered due to being exposed to damage-associated molecular patterns as hepatitis has been ongoing, so that these cells start to express toll-like receptor 4, complement receptor, immunoglobulin receptors, as well as other factors that will make the environment, the local environment, including the LSEX, more susceptible to for cells that enter the circulation to cross into the liver. And this will have profound changes, not only to the activation and the ongoing inflammation that will occur in the liver, but also the spill effect that will occur when these changes spill over into the systemic circulation. Recently, a macrophage population was identified in the setting of using single cell technology that was characterized as TREM2 CD9 positive cells that were associated with a fibrotic niche that develops. And I'm sure that ongoing research and collaborations in the future will help us to also look at the inflammatory niche, which is typical for ACLF. So if we look at specifically what happens in adipose tissue, we have previously shown that as patients with non-alcoholic fatty liver progress to non-alcoholic steatohepatitis and fibrosis, a profound change occurs also in the adipose tissue environment. We know that normal adipose tissue will be characterized by adipose tissue, resident adipose tissue macrophages that are normally CD11b positive cells. But we could show in the presence of NASH and NASH with fibrosis that adipose tissue macrophages undergo profound changes, so that they now express CD206, express CD11c, and is also that they are also capable of producing into culture environment chemokines, microRNAs that are released into the portal vein and the systemic circulation. And we at the expression level could show that in the absence of adipose tissue macrophage activation, that NASH did not occur. So it seems as if using RNA sequencing, that it seems as if the development of NASH, that adipose tissue macrophage activation and inflammation was a prerequisite for the development of NASH. So that the take-home message really is that in adipose tissue, profound changes occur. They form many different types of niches that involve different types of macrophages, and some of the inflammatory macrophages seem to be responsible for the production of cytokines. In a recent beautiful study, it was also shown that CD11c positive macrophages are also capable of releasing chemotaxins, including CXL14 and CXL16, which will then be responsible for recruiting neutrophils to the liver. So this is, I think, a very exciting finding, because it shows that adipose tissue actually affects what happens in the liver by what they produce and what they secrete into the portal circulation. Now, a very important aspect of our research, and the research also done in Spain by various groups, including Augustine, I think, is related to the intestine. I think what we know is that under normal circumstances, the intestinal macrophages are normally inert to stimuli from bacterial products, so that these cells are dedicated cells in terms of phagocytosis, but they do not release pro-inflammatory cytokines under normal circumstances. And this happens because of the barrier that is protecting cells from bacterial translocation, excessive bacterial translocation, dendritic cells that monitor the environment, and that because of the barrier, the mucus layer, and signaling that occurs under steady state circumstances, and the secretion of antimicrobial peptides by Paneth cells, this environment is maintained to be anti-inflammatory. Previously, in 2013, we showed taking biopsies from patients with decompensated cirrhosis, compensated cirrhosis, and controls. By taking small bowel biopsies, we showed that the local environment in these patients changed. With an influx of macrophages into the intestine that now express CD14, TREM1, and INOS, and we could show that these macrophages secreted IL-6, nitric oxide, IL-8, and CCL13 into the culture supernatant. So in patients with cirrhosis, this niche completely changes, and from a tolerogenic IL-10 background to an inflammatory background, without that being obvious when you look at tissue slices under the microscope. So this is what we first observed back in 2013. At that time, not knowing what this meant, we saw the influx in compensated and decompensated cirrhosis of plates of INOS-producing cells that were highly significantly different from the steady state condition. And also, we could answer at that time the question, because it was still proposed that due to portal hypertension, they may form pores or openings that may allow bacteria to translocate. But by studying this under electron microscopy, we could show that the tight junctions were not different. The gap junctions, the desmosomes, and the adherent junctions were not different in patients with decompensated cirrhosis compared to controls, suggesting that that is not the mechanism by which bacteria cross. When we looked at the immune cell populations using co-localization studies, we could show indeed that the cytokines were produced by CD68 positive and INOS positive cells, and that the macrophages were indeed responsible for the change in the local environment that occurred. This also resulted in a decrease in transepithelial resistance, meaning that the membrane, that the gut became more leaky, allowing certain macromolecules to cross into the circulation. So if this is the case in 2013, I wish that I knew that I was in for a very, very long and difficult ride, because we realized at that time that using biopsy material from patients would be insufficient to really understand the changes that occur at the level of the gut. And also following the publications by Augustine and other groups, it really became apparent that it is not an easy focus area for us to be involved in. And that really, it took a long time for us to move on to the next phase because of the difficulty. So what became apparent during the development the last few years, and also from publication from our group, was that we should view macrophages in the intestine differently. We should view them as macrophages that may originate from monocytes and infiltrate, even under normal circumstances, the intestinal lamina propria, where they will support the epithelial barrier and where they will be taking part in phagocytic and bactericidal activity. But we've also come to realize that in the intestine, there is niche-specific macrophages. This was first characterized a couple of years ago by the Italian group when they described the gut vascular barrier, but with subsequent work also from our group, it was shown that there are niche-specific macrophages. These are long-living blood vessel-associated macrophages that support the integrity of the vascular barrier and prevent vascular degeneration as well as increased vascular leakage. So this has become the focus in our lab to see, can we understand this process during the development of cirrhosis in this animal model. So we propose that loss of blood vessel-associated self-renewing macrophages in the gut may represent the key mechanism underlying the impaired gut vascular barrier, allowing translocation to occur. So this was the hypothesis that we generated. In terms of creating the ideal model, I think it is, we certainly wanted to focus on a model that was stable, that could recapitulate cirrhosis to an extent that it was acceptable. And we knew that from our experience also in the literature that not any model that at that time was available really did that. So we went about by using a CCL4 model, but instead of it being an aggressive model with cirrhosis being induced at 8 to 12 weeks, we rather used and induced a protracted model, dosing between 2 and 3 times a week for a period of 19 weeks with 1 week washout period. And we could reproducibly show that using this model, that at 14 weeks, we could show advanced fibrosis. At 17 weeks, this was even more advanced. And at 20 weeks, we could show a model that really simulated micronodular cirrhosis to some extent. So this was quite important, as we know that the model is ultimately going to be useful when interpreting your results. So the second thing that we developed was a inducible reporter mouse model where we used tamoxifen to activate the CREA recombinase enzyme, enabling us to insert YFP as a reporter, enabling the expression of YFP positive cells in CX3CR1 macrophages. From this, the understanding is that only self-renewing macrophages will express this YFP reporter, whereas all incoming macrophages will be negative to this reporter. So by generating this model and then by going on to expose these mice to CCL4, we now have a reproducible model of advanced fibrosis and cirrhosis, where we can show that only long-lived self-replicating and self-maintaining macrophages are YFP positive. That allows us to study, because it's these macrophages that are responsible for maintaining the vascular barrier, and we have therefore been able now to develop a model that allows us to see these macrophages in more detail. When we look at the liver in these patients, in these mice, we could show that at 19 weeks, and this is 8 weeks, that changes occur when we looked at the expression of CLEC4 in the livers of these animals to confirm that macrophages indeed at 19 weeks are changed. They co-localize CLEC4, meaning that these macrophages are activated by very likely PAMPs and bacterial products originating from the gut, showing us the validity of our model. So by using this model, the CRE model, and by inserting the reporter, we could very nicely show that at 14 weeks, that the macrophages, at 14 weeks, we could still show macrophages. So in this case, the IB, the long-lived macrophages will be macrophages, the yellow macrophage, yellow-green macrophages, and the IBAR1 macrophages are the incoming macrophages. And here one can see by inducing CCL4 that by 14 weeks, we still see that the macrophages in the control animals are nicely localized around blood vessels, as we expect them to be at 17 and 20 weeks. But in the CCL4 model, we see that after 14 weeks, the YFP positive self-renewing blood vessel-associated macrophages that should maintain the gut vascular barrier are lost. And this very nicely correlates with the presence of fluorescein in the plasma when we did isolated ileal loop experiments in these animals. So by doing this, we have found a reproducible model. We are currently at time point 14 and time point 19, so 20 weeks. We are currently evaluating these cells using SmartSeq and to see in the control and CCL4 exposed animals if we can identify pathways that are unique and that we can utilize in order to better understand what needs to happen to those cells when the vascular barrier fails. So I hope that I've convinced you thus far that adipose tissue is important. I think that the gut vascular barrier is of incredible importance, very difficult to study, but that we have now found tools that we think may help us to optimize how we are going to assess the vascular barrier in the future. And in the next part of the talk, I will focus on the peripheral circulation and specifically how it relates to the development of acute on chronic liver failure. So I'm preaching to the converted, and as you know very, very well, that ACLF is a very dynamic process, often involving a precipitant in patients with cirrhosis that allows these patients to decompensate so that organ failure develops with high associated mortality, and that there is the coexistence of two factors, that there's evidence of immune activation if you purely focus on the peripheral circulation looking at cytokines, but that there's also a suppressive state so that immune dysfunction in these patients somehow is very closely linked to hepatic failure. So what is also important is that ACLF does not occur from the one day to the other. It is a disease that very likely starts decades before, when due to perpetual liver inflammation, there is priming of the immune response, as I've shown you from the beautiful publication from Augustine, where he nicely showed that in 2014. So when immune function develops, we can say that it's compartmentalized, it's compartment specific, it's filled from the gut. It is perpetuated by sterile and infectious events that release BAMPs, DAMPs into the circulation to react to these pattern recognition receptors to maintain this inflammatory state. But finally, when most likely through many mechanisms, immunoparesis will develop, that makes these patients incredibly susceptible to infections. And this is what I try to show in this slide, that in the presence of this perfect storm, ongoing inflammation in the liver, a gut barrier that fails, there will be circulatory changes that occur, either through portosystemic shunting, that bacterial products entered into the circulation, or through spillover effects from the ongoing inflammation in the liver. But what is important is that we recognize in the systemic circulation various factors. We recognize that there is increased levels of pro-inflammatory cytokines that correlate with disease outcome. But if we analyze the cell types, we now see that profound changes occur with the expression of immunomodulatory cell populations that include MERPTK positive cells, macrophages that express AXL, but also monocytic myeloid-derived suppressor cells that can be recognized in the circulation. What is incredibly interesting is that it has now been shown that neutrophils can only cross into compartments with difficulty. So if an infection occurs in the peritoneum, it is more difficult for neutrophils to reach the site of infection. Whereas these immunomodulatory cells can move in and out of of compartments where they then would modulate the immune responses. Normally, these cells will be involved in tissue restoration, but if the process is ongoing for long periods of time, these cells will be responsible for a reduced capability to defend against bacterial products and favor, instead of immunopathology and organ damage, will favor the development of bacterial infections. So we started to ask the question, but we now know that these populations exist in the population, but what makes them different? The previous studies only focused on describing some elements of these cells, but could we also at the expression level understand much more of how these cells are different? So what we then did is to analyze using flow cytometry and RNA sequencing the expression profiles of these cells in the circulation of patients with ACLF, with decompensated cirrhosis and controls. And what we found was what was already known, is that there is a decrease in the expression of HLA-DR, which is important in especially antigen presentation. But this has been known for a long time. But what we in addition found was that there was a decrease in the classical monocyte, the frequency of classical monocytes in the circulation. These are the CD14 positive, CD16 negative populations, as well as a profound expansion of the intermediate cell population, the CD14 positive, CD16 positive population of intermediate monocytes. And what is very striking is that it is indeed these cell populations that produced interleukin 10 and also associated with classical monocytes now being able to produce and secrete interleukin 10 into the circulation. So we could conclude that there's a decrease in CD14 in classical monocytes, increase in MERTK, with increase in IL-10 producing circulating monocytes that contributed to the production and the release of IL-10, an immunosuppressive anti-inflammatory cytokine, into the circulation. When we then looked at the RNA expression level at genes, we found the following, and I will take you through this slowly because it is a busy slide. So what you can see here is volcano plots in red of genes that were upregulated, in green genes that were downregulated, and the 10 most important pathways associated, biological consequences associated with the genes that were differentially expressed between decompensated cirrhosis and controls, acute liver failure, acute on chronic liver failure and controls, and ACLF versus decompensated cirrhosis. And what we could show was, if we compared decompensated cirrhosis patients to controls, we could show that even in a decompensated cirrhotic, there was still the ability to mount effective immune responses, or at least some immune responses. If you look at the number of genes and the biological relevant pathways, these upregulated genes in decompensated cirrhosis compared to controls were associated with immune responses, immune system responses, regulation of T cell activation of leukocytes, etc. So there's some degree of still the ability to mount immune responses in the monocytes of decompensated cirrhotic patients. When we then looked at the ACLF patient population versus controls, we found that the opposite. We found that the most importantly downregulated genes, when we compared ACLF to controls, were gene profiles and biological processes associated with leukocyte adhesion, with cell activation, and with some components of immune cell activation. And this was even more pronounced when we compared ACLF with decompensated cirrhotic patients, because here we can very nicely see that comparing ACLF to decompensated cirrhosis, that they were predominantly downregulated genes and biological processes involved in the regulation of immune responses when compared to decompensated cirrhosis. So we could conclude that there's a transcriptional switch. There is an involvement of multiple components, and that ACLF is clearly a distinct phenotype when we compare that to patients with acute decompensation. And just again, focusing on this in ACLF versus decompensated cirrhosis, how clearly downregulated the genes were across the spectrum of inflammation responses between ACLF and decompensated cirrhotics. So clearly a different disease entity, and there should be no doubt about this anymore. So what are the functional consequences of this molecular signature? Well, we could show that in ACLF patients compared to controls, that toll-like receptor 2 and 4 expression was reduced. So that already shows us the impaired ability to recognize components of bacterial products, including LPS. And when we cultured these macrophages, the CD14 positive macrophages, and looked specifically at the ability to phagocytose using whole blood phagocytosis culture assays, phagocytosis assays, we could very nicely also demonstrate that it's not only at the transcriptional level that these cells are different, but also at the functional level with the reduced where the macrophages, CD14 positive monocytes from patients with ACLF had a reduced capacity to phagocytose E. coli, as well as a reduced oxidative burst response. So when we looked into detail at the number of genes involved, as well as the biological processes, we found that many of the genes that were involved in important processes when phagocytosis and internalization of the bacteria occurs before reactive oxygen species can be formed were downregulated. Important genes like transcriptional factors, IRF8 was downregulated, as well as important genes that were necessary in the assembly of the units to be transferred into the phagosome for the release of release and the formation of reactive oxygen species, as well as upregulation of genes associated with a blocking effect of this process, so that we could conclude that not only at the expression level, at the functional level, but also based on various genes affected in the pathway, was there a plausible reason why phagocytosis was impaired in patients with ACLF. We then looked to some extent at the metabolism, because it is well known that macrophages and monocytes that during an inflammatory so-called M1 response, that they rely on glycolysis for their energy demand, and when macrophages are anti-inflammatory or M2-like, we would expect to see a change for these, because these macrophages will, rather than utilize glycolysis pathway, switch to a fatty acid oxidation pathway. And we focused on our cell populations to see to what extent we can show differences. What we could observe was that there were various genes involved in the glycolysis pathway that were downregulated in our patient cohort. And we then reasoned, but is it not possible, if we can increase the substrate into the Krebs cycle, could we not improve macrophage function? And what we did is we designed an experiment where we blocked the export of glutamine out of the Krebs cycle and increase the substrate utilization of glutamate. And what this resulted in was that in macrophages, monocytes exposed to MSO that blocked this pathway, we could show that there was an increased, we could partially restore phagocytosis by this blockage of and import of glutamate into the cell cycle. And we could also decrease the expression of IL-10 at the expression level and increase TNF-alpha, restoring the pro-inflammatory phenotype of the monocytes. So what are the most likely factors acting as precipitants? Well, first, I think it's important because in our article that was published a while ago, a lot of emphasis was placed on the experiment using metabolism. But I think what was really missed from that publication was what we could show also in that publication, and that was that when we took ACLF plasma from patients where we pulled ACLF to patients' plasma, and we then used 20% volume for volume conditioned culture experiments exposing healthy monocytes from healthy individuals to ACLF plasma or to normal plasma, we could show that we could induce a phenotype in healthy monocytes that resembled very strongly the phenotype that we observed in ACLF monocytes that we obtained from ACLF patients. So this was, I think, remarkable, both recapitulating the pro-inflammatory changes and the anti-inflammatory changes very, very nicely. So this told us that in the plasma of a patient with ACLF, that a circulating factor must be present that allows the induction of a phenotype to resemble the ACLF phenotype in a healthy monocyte. So what this also showed was that by inducing this phenotype, we could show that we could reduce TNF-alpha expression, IL-8 expression, and that we could increase IL-10 and MERTK, really very much resembling the ACLF phenotype that is observed in ACLF monocytes. But more importantly, also, it's not just at the expression level. If we cultured these monocytes and looked at the ability to phagocytose E. coli over a period of 36 hours, we could very clearly see that not only are these cells affected by ACLF plasma at the expression level, but also functionally. And this is not a toxic effect because only after a few hours does this deviate, showing that this is not a toxic effect. This is actually a change in phenotype induced by the co-culture experiment. So the key message is therefore that ACLF plasma induces an ACLF-like phenotype in healthy monocytes that resemble the expression profile that we observed. This confirms that circulating factors induce this phenotypic switch, and that this affects the cells at the functional level. So I think there's a lot of evidence in the literature that already looked at various factors that may be responsible for precipitating ACLF, and that are circulating factors in the circulation, including very nice work from Juan Claria that showed that oxidized albumin is responsible for increasing mRNA and protein expression in leukocytes, especially affecting the increase in interleukin-1 beta, IL-6, IL-8, and TNF-alpha, and also upregulated the expression of COX-2, prostaglandin E2, and prostaglandin F2 alpha, which shows that oxidized albumin, which occurs in patients with cirrhosis and decompensated cirrhosis, affects the functioning of circulating monocytes profoundly. and also recently by, again, beautiful work from the Spanish groups showing that albumin also modulates endosomal toll-like receptor signaling. And this is when normal healthy albumin is administered, it does influence the way that toll-like receptors are processed in the cell. So the molecular and functional signature of progression recovery, I think, will help us to understand how, because what we have done is certainly very interesting work. It is, however, descriptive to some extent. And I think in following studies, we need to move on to more work looking specifically at mechanisms of progression or mechanisms of recovery. So together with a PhD student in our lab, we have designed this protocol where we take patients with alcoholic liver disease that develop acute and chronic liver failure, or that develop acute decompensation, but not yet fulfilling the criteria for ACLF. And we then looked at patients that we followed all of these patients for a period of 28 days. And we looked then at collecting samples at day 0, 3, 7, 14, and 28. And then we looked at when we stratify these patients according to patients that progressed or required transplantation versus patients with ACLF that recovered, could we identify a signature that helps us to show what is necessary for a patient to recover. And the same with patients with acute decompensation cliff score of more than 60. So these are not just patients with a cliff of more than 50, because we found that they never progressed, but we could show that these patients actually do progress. And again, here we wanted to identify the phenotype of patients that progress to patients that never progress. So although we are still evaluating, because the study has generated enormous data that is now analyzed, but what I can share with you, that it seems that if in the non-progressor over time, it seems that if phagocytosis, so the functional ability of monocytes must recover in order for a patient to not progress, or for an ACLF patient to progress further. So we are now analyzing, and I hope to share with you the data soon of the pathways that are necessary, and that will help us to depict the progressor from a non-progressor at a molecular level. So in the last 2 slides, I would just like to spend a little bit of time, if I can, just to share with you what I have shown, and I think where we stand with this, because this is also based on enormous work from various groups, so that I think we can conclude that if there's ongoing inflammation in the liver, Kupffer cells are activated, LSECs are activated, allowing cells to move into the liver. And this process, if ongoing, will lead to various consequences. The active inflammation state in the liver, but also outside of the liver in various tissue compartments, the development of portal hypertension, with the consequences of portal hypertension, the fact that vasoactive substances are produced and are at maximum, so that in the presence of an infection that disrupts this precarious state, that this balance will tumble, allowing hypotension to occur that cannot be corrected, allowing organ hyperperfusion and organ dysfunction to develop with the development of multi-organ failure. And this is fueled by bacterial infections and PAMs from the circulation, from the intestine, and also bacterial translocation from the gut. So I think that there is a clear need also to understand better the interaction between what happens at various levels and various compartments, and how these interactions occur with the consequences, especially in this phase when organ failure is so advanced. So in the last slide, I think ACLF is the absolute perfect storm. Various compartments contribute. The production of inflammatory cytokines, PAMP, STAMPs, lead to systemic inflammation. that through cytokine release or immune exhaustion, either precipitated by infections that leads to changes in the circulation, or by infections or alcohol binge, will lead to hepatic decompensation and immunoparesis. And that is the perfect storm that will lead to the development of organ failure. So in conclusion, the distinct transcriptional profile in ACLF is that of an immunotolerant state with profound consequences. There seems to be changes in glycolysis and fatty acid metabolism that affects how these cells perform, and we can, to some extent, correct this by introducing substrate into the Krebs circulation. Ultimately, however, immune dysfunction seems to be intrinsically and decisively coupled to hepatic failure, and how this association works, I think we still need to discover. Then I would like to thank all of my PhD students in the lab, and also my molecular biologist, Hanni Korf, which has done tremendous work that I've shown you today. And then I would like to thank you for your attention. It is a very important question that I'm not sure I know the full extent of the answer. What we know is that that they are very important in maintaining the vascular barrier, because if they are deleted either through infection or through specifically targeting these cells, increased bacterial translocation occurs and the vascular changes. The vasculature can be on a microscopic level will change. These blood vessels become abnormal. So how this exactly works, we are not 100% sure, and this is still why we are awaiting the single cell analysis data, because I'm sure we will be able to deduce from that the answer that you are getting at. But that they are actively contributing to the maintenance of the vascular barrier has now been, I think, accepted. The understanding that I have at the moment, based on our work, was what was amazing to me is that even in patients with quite profound decompensated cirrhosis without ACLF, that at the transcriptional level, they were capable of mounting immune responses. And that if you compared them to healthy controls, you could clearly see that. But as ACLF develops, that really completely changed. And what I showed with the volcano plot and the comparison between ACLF and decomps was that clear downregulation of immune response genes in the ACLF patient. So I guess that it really comes down to probably two factors. That over time, as these patients are exposed to prolonged prolonged periods of inflammation, PAMPs, DAMPs, and other factors, that immunotolerance may develop, and that immunotolerance in these patients may be epigenetically modulated, meaning that this is required, and that it happens in response to trying to preserve the host from developing organ dysfunction and prevent immunopathology. So my understanding still is based on the fact that I think immune tolerance is something that we took over from the sepsis literature, where we know that if you expose them, that they will expose monocytes or macrophages, that they will over time decrease in terms of their ability to respond to stimuli. So the epigenetic changes associated with that was also recently studied by Thierry Gusteau's group, but I'm not sure that we fully understand that yet. As far as I know, it has not been study in that way, Rafa, but what we have done was to try to prevent that from occurring, that there would be problems with interpreting the data by including only patients with ACLF grade 2 in our analysis. And the rationale for that was, if we are going to include patients with ACLF grade 3 or patients with pronounced organ dysfunction, we may start to see profiles that are not related to ACLF, but rather to organ failure or sepsis. Also, if we're going to include ACLF1, ACLF2, the phenotype may not have been that distinct that would have allowed us to contrast that to decompensated cirrhosis. So I would expect that is going to be the case, that you would see with progressive disease that the expression profiles will be more severely affected. And we have some data suggesting that, because in the Burns-Meyer paper, they did show that the MIRC-TK expression correlated with MELD scores in their study population. So I would expect that, but I think it is difficult because it is a heterogeneous disease, as you say. I think it is important that if we study this, that we try to keep it as homogeneous as possible. So we only included patients with alcoholic liver disease. And then we only focused on ACLF grade 2, as I said, because of the concerns that we would not be able to get a clear signal if we're going to include all ACLF. It's a very good question. No, we didn't do that. But I think that would be necessary as well. We didn't expect that at the time, and we had to make certain decisions regarding on how we're going to spend our money. But it was no, we didn't do it, and I think that's a very good suggestion. I'm actually very enthusiastic about the possibilities. Because I think it is now possible. It's now possible to target AXL. It's now possible to target TOM. It's now possible to target MIRC-TK selectively. But one interesting concept is the use of metformin. Metformin, for instance, interferes with AXL on macrophages and will reverse the function of the immunomodulatory function of AXL-positive monocytes in the circulation. The problem, I think, that we will have in the coming years will be to understand the off-target effects of these drugs. And there are now monoclonal antibodies that we can manufacture and utilize to test this, and also drugs that we can test. But the biggest issue and concern would be, if we test this in patients, will be the off-target effect. So it seems as if in cell culture systems ex vivo, it works pretty well, and we can change the functions and the expression profiles of these cells. But we will have to see, and we will have to be very careful when we design trials to look at what will happen in patients, where we may see that by switching off AXL or other immunomodulatory cells, that we may worsen inflammation that may also be detrimental. So I think that's going to be the tricky part. Very much.
Sure.
See you soon. Bye-bye.
Again. Bye-bye.